Hi.
Hello. How are you? Good, yeah. Good. Are there any exciting new stuff in?
Not in my talk, because I heard it was being live streamed.
Can I just introduce you to Jodrell, Professor?
Jodrell. Yeah, Jodrell.
Still 1 minute.
1 minute.
Do you want to start right at 3 or do you want us to wait a little bit longer?
Entirely up to you. Whatever you normally do here. How many people are there in the DRI?
There are about 350 researchers. Well, the
Okay,
entire
most of them are.
They're all going to listen.
They're all going to listen. Yeah, right.
All going to steal your. I'm just kidding.
Well, let's see. Hi, Bart.
All right, so let me introduce, well, a guy who doesn't need an introduction, Professor David Attwell. He's a Jodrell Professor of Physiology at UCL. And as you know, his lab has been focusing quite a bit in really understanding And I think he's one of the pioneers, he has been one of the pioneers in the field for a very long time, where even before glial cells became fashionable and hot, David has been studying and finding very interesting and very important contributions in understanding how these different cell types, including pericytes, which are the cell types that you'll hear a lot about today, and neurons work together to maintain brain homeostasis. And so today I'm very excited that he's here to share with us a lot of the stuff what he has published recently in Science, but he'll start with really with giving us an overview of the role of pericytes, capillary pericytes in health, and then he's going to talk about ischemia and AD models. One thing I want to emphasize is that there are going to be a lot of listeners online listening through the YouTube, and for people who are listening there right now, please, you're welcome to, if you have any burning questions, please tweet me. It's a modern day now, people tweet. So please tweet me at Soyan Hong Lab, or email me at ucl.ac.uk for any questions. And David, it's all yours.
Okay, thanks for the invitation and the introduction. So today I'm going to try to persuade you that these little red cells here on capillaries are crucial controllers of energy supply in the brain, both normally, in ischemia, and in the last half of my talk, in Alzheimer's disease. So I'm going to start out by looking quite generally at why and where brain energy supply is controlled. I'll then show you that pericytes can constrict and dilate capillaries really quite profoundly. And I'll look at the mechanisms by which they do this. It's largely controlled by calcium. And so I'm going to look at calcium sources for this constriction and also the synapses, which regulate the tone of pericytes. I'll look briefly at the role of astrocytes, and I'll show you just briefly that there are different signaling pathways regulating brain blood flow at the arterial level and at the capillary level. And then I'll kind of turn to the dark side, and first of all look at the role of pericytes in ischemic pathology and how they play a major role after stroke, and then look at Alzheimer's disease and finish off with some therapeutic approaches, in particular for AD. So let's start out by looking at why and where brain energy supply is controlled. I'll start out with why. If we look at the brain of a typical Brexit voter, you'll see that it's fixated on the things that Brexit voters are fixated on. And Homer's brain is 2% of his body. Would it help if I put the lights down slightly? Homer's brain is 2% of his body, but it's 20% of his resting energy goes to power his brain. So as you're sitting here, 20% of your lunch is powering your brain at the moment. And in fact, in children, it's even greater. It's about 60%. And that's why we have this dense network of blood vessels bringing oxygen and glucose into the brain. So many of you will be familiar with hippocampal slices. These little bobbly things are the neurons, but you'll be less familiar with the fact that for this bit of the brain to work, it requires this dense plexus of blood vessels. But even then, not all the neurons can be firing at a maximal rate at the same time. So if a part of the hippocampus up here wants to think about something, where we are in the world, for example, then it has to dilate local blood vessels by releasing vasodilatory factors to bring in more oxygen and glucose to power those neurons. And this dependence of the brain on energy supply and its crucial need to increase local energy supply when it's thinking about something underlie the fact that energy disorders play a major role in lots of neurodegenerative conditions. So where is the brain energy supply controlled? Here's a simplified diagram of the plumbing of the cortex. Blood comes down from the surface of the brain, the pia, through penetrating arterioles and goes into our capillaries where nutrient exchange occurs with the neurons. And it's generally been assumed that most of the regulation of this energy supply, the flow of blood, is done by smooth muscle cells wrapped around arterioles. But it turns out this is not the whole story, because these old guys, Karl Ebert in Germany and Charles Rouget in France, discovered these cells called pericytes located on capillaries, many of which are contractile. And they were named 50 years after they were discovered, and they have at least 5 functions. They're actually found on blood vessels throughout the body. They're involved in the formation of blood vessels. They maintain the blood-brain barrier. After pathology in the brain, like spinal cord injury, they help to form the core of the so-called glial scar, even though they're not glial cells. They regulate immune cell trafficking in the brain, both entry across blood vessel walls and also movements once they've got into the brain. And finally, and what I'm going to talk about today, they control capillary diameter. So I just want to spend a minute to illustrate the rise of studies on pericytes. So they were discovered, as I said, hundreds of years ago. Nothing much happened for 100 years, but now there's been an exponential rise in the number of papers on pericytes. So every day, about 1.5 papers are published on pericytes. But nevertheless, they remain very poorly understood. They're a major cell population in the brain. Every 30 to 50 microns along a capillary, there's a pericyte. But if you go to PubMed, you'll find that only 1 paper on the brain in 1,000 mentions pericytes. And it's even worse in the heart. In the heart, pericytes are the second most common cell type after endothelial cells. So the contractile cells in the heart, the myocytes, are an almost irrelevant third. But only 1 cardiac paper in PubMed in 5,000 mentions pericytes. So we're clearly missing out on some therapeutic opportunities because pericytes contribute to all diseases involving ischemia. That includes stroke, heart attack, spinal cord injury, as well as Alzheimer's disease, as I'll show you today, and also diabetes and cancer. But they're largely ignored by scientists and big pharma. So I'm a kind of missionary for pericytes, trying to persuade you to study them. This is what they look like. This is in the cerebellum. This is in the retina. The green label is isolectin B4, which labels the basement membrane, and so it outlines the capillaries. The pericytes are labeled with antibody to the proteoglycan NG2, which they express. So these red cells on the straight parts of capillaries and at their junctions are the pericytes. And if you look at them closely, you see they put out long processes along vessels and around vessels if you patch clamp them. So they seem designed to cuddle a vessel and squeeze it. They seem designed to be able to adjust the diameter of capillaries. And it's interesting then that Edith Hamel has shown that 2/3 of the noradrenergic innervation of the vasculature in the brain ends near capillaries, not near arterioles. So maybe that's telling us something about the locus of control. I want to show you in an artificial situation, first of all, that pericytes can profoundly adjust blood flow. This is a capillary in the retina where you can take an isolated retina and get long lengths of capillary, which are all in focus. If you then push a patch pipette up against a pericyte, which is hidden under here, and depolarize it, then you find that when you pass depolarizing current, it completely closes off the capillary, as you can see here. So they have the power to completely occlude blood flow. And if I play the movie again, I'll show you another remarkable thing, which is that pericytes talk to each other. So that after this one constricts, you'll see that this one constricts. So this one constricts now, and a bit later, this one constricts. And you can see this by plotting the diameter at the locations of pericytes here. This is the central stimulated one. These are the ones on each side. They repeatedly constrict. But nothing happens in the endothelial tube in between. So pericytes are the only contractile cells on capillaries. This is just a kind of in-principle experiment to show you they could regulate blood flow. That talking to each other is believed to occur by virtue of an electrical signal passing along the endothelium from one pericyte to another, as was shown some time ago by Don Puro's group. This constriction of pericytes is calcium dependent. And when we get on to disease later, you'll see that it's malfunctions of calcium concentration which lead to the deleterious effects of pericytes, both in stroke and in AD. So if you repeatedly stimulate one of these pericytes and evoke a reduction of capillary diameter, as you can see here, if you take away the external calcium, 2 things happen. First of all, you abolish the constriction, and secondly, you get a poorly maintained dilation, as though there's some endogenous tone being produced by the pericyte. So the constriction totally depends on calcium. So I want to look at the sources of calcium and also the synapses which regulate calcium so that you can understand how they're controlled normally. Let's look at calcium sources first. Here's a pericyte with its arm wrapping a little capillary. There are myosin and actin within it. What I just showed you was that if we depolarize the pericyte with current, then that opens voltage-gated calcium channels, calcium goes up, and that activates myosin light chain kinase to activate the myofilaments to constrict, so the capillary gets squozen. Okay? Of course, this is not how things happen in the brain normally. Quite often, calcium is raised in cells by G protein-coupled receptors. So if we apply a vasoconstricting agent, endothelin, which is going to become very important in the Alzheimer's section of the talk later on, and we measure the calcium in pericytes, this is in a brain slice, then when we apply endothelin, we find there's also a rise of calcium concentration, as you can see just here. And if you measure the diameter of the capillary at the same time, you see that it decreases when you apply the endothelin. Okay, so endothelin, which is an endogenous agonist for vasoconstriction in the brain, has the power to decrease capillary diameter. Now, how is that working? Well, the pathway in this case is as is shown here. And this will be important when I come to therapeutic opportunities later. Endothelin acts on endothelin A receptors in pericytes, releasing inositol trisphosphate, which dumps calcium from internal stores, and that again activates the contractile apparatus, as I just showed you, for depolarizing the cell. But it turns out it's more complicated than this, because the endothelin itself only raises the calcium a little bit. And the rise of calcium which occurs is amplified by another mechanism, which we haven't published yet, which is that this rise of calcium activates a calcium-gated chloride channel called TMEM16A, and that the chloride gradient is depolarizing in these cells, like it is in GABAergic neurons early in development. So chloride goes out, and that depolarizes the cell, and that leads to opening of voltage-gated calcium channels, and the calcium elevation is amplified. And we know this is happening because if we apply endothelin, here's the constriction produced by endothelin, and we then apply two structurally different blockers of this chloride channel here, called Ani9 or MONA, both of these greatly decrease the amount of constriction which endothelin produces. And in other experiments I don't have on the slide, we've shown that that decreases the calcium elevation as well. So endogenous GPCR agonists can release calcium from stores and evoke constriction, but that's amplified by this combination of the calcium-gated chloride channel and the voltage-gated calcium channels. Okay, so where is blood flow control initiated? Say you're a neuron and you want to think about something. How are you going to increase your local energy supply? Well, Thelma Lovick has shown, at least in hippocampus, that most neurons are closer to pericytes than they are to arterioles. They're about 15 microns away from pericytes, but about 100 microns away from arterioles. So if active neurons or their associated astrocytes release a diffusible messenger, it's going to reach pericytes first. So you might think that active neurons would signal to pericytes. Then by the electrical conduction mechanism that I showed you earlier, that may propagate between pericytes, and it could even propagate back to the upstream arteriolar smooth muscle. So we want to know whether pericytes can also be constricted or dilated by messengers released from neurons or glia. I've shown you endothelin already, but I want to show you some synapses which can regulate this now. And this matters because we want to understand normal control of blood flow in the brain, what goes wrong in pathology like AD, and also how functional imaging signals are generated. Because as you know, there's an enormous cottage industry of psychologists studying the brain by getting images of little red dots superimposed on structural images of the brain. And it's very poorly understood exactly how these are generated. These result from an increase of local blood flow, which is somehow triggered by neuronal activity, bringing in blood with less deoxyhemoglobin in it. So what you're seeing with this red blob is the fall of deoxyhemoglobin concentration. And by the end of this talk, I hope you'll go away in a frame of mind wherever you see this in future, you'll think, yeah, pericytes, because that's what's generating this. So what triggers the increase in blood flow that generates bold signals? So what are the synapses which might regulate blood flow in the brain? So if we apply, I'll show you a movie of this in a second. If we apply noradrenaline to capillaries, we find that the capillary diameter goes down at pericytes. And if you superimpose glutamate, you find the diameter goes up. The glutamate mimics neuronal activity, also evokes neuronal activity. So here's a movie of this happening in the molecular layer of the cerebellum. This is the molecular layer. These are red blood cells in a capillary. There's a pericyte sitting just here. And when I play the movie, if we apply noradrenaline now, you'll see that this pericyte squeezes the vessel, and when we superimpose glutamate, it dilates the vessel. So these neurotransmitters can constrict and dilate capillaries by acting on pericytes. How does it work? What generates the capillary dilation in particular? Because it's the dilation you need to bring in more energy, both to generate bold signals and to power the neurons. So here's a diagram of a typical glutamatergic synapse. 85% of the synapses in your brain are like this. When action potentials come along, they release glutamate. That gates postsynaptic glutamate-gated ion channels. Ions go in, and then the glutamate is recycled. Now, if you do an energy budget for the brain, as Simon Laughlin and I did about 15 years ago, 20 years ago, then you find that if you add up the ATP used on pumping out all of these ions postsynaptically and also presynaptically on the action potential and in the glial cells, then synapses use most energy in the brain. So about 60% of the energy used by your brain goes on synapses, especially the postsynaptic side of synapses. And so it's appropriate that it's synapses which release the messengers which can dilate blood vessels. And this happens as a result of calcium elevations, both in postsynaptic neurons. For example, NMDA receptors raise calcium, and that can activate enzymes like nitric oxide synthase and phospholipase A2 or D2. And in glial cells, it used to be thought the main raiser of calcium was metabotropic glutamate receptors. I'll show you in a second it's actually ATP receptors in this scenario. But both of these enzymes generate vasodilatory messengers. Phospholipases make arachidonic acid, which are converted into prostaglandins. Nitric oxide synthase makes nitric oxide. And these can act on pericytes or indeed on vascular smooth muscle to relax the myofilaments and generate a dilation. And this is done in the context of a background constriction produced by noradrenaline. So how does this work at the capillary level? We can look at this by patch clamping pericytes. So here we have transgenically labeled pericytes with DS red telling us which cell is which. We can use that to patch the cell. We put a dye in the pipette so we know we've patched the right cell. And if you apply glutamate or NMDA or activate axons to release glutamate endogenously, then you get an outward current produced in these cells. This is very different to what you'd see if you apply glutamate to a neuron normally. Normally, glutamate activates AMPA or NMDA receptors, and at the resting potential, we get an inward current as sodium or sodium and calcium go in. But here we're getting an outward current, and this reflects the activation of a potassium current. Okay? So what's going to happen in practice when you get this outward current generated? Here we're stimulating the parallel fibers in the cerebellum. You're going to get a hyperpolarization of the pericyte by about 10 millivolts. And because the resting potential of the pericyte is about minus 40 millivolts, it's right in the activation range of L-type voltage-gated calcium channels. As you go more negative in voltage, those calcium channels are going to close, so there'll be less calcium coming in, and the calcium concentration will fall. And that ought to produce a relaxation of the myofilaments. So can we see this? In fact, we can. If we image pericytes in vivo with GCAMP expressed inside them. If we look at pericyte processes, this is pericytes on the first to third order of the capillary tree. If we stimulate the whisker pad and image pericytes through a cranial window in the somatosensory cortex, you get a fall of calcium concentration, as you can see here. And you would expect this to generate a relaxation of the pericytes to increase their energy supply. And you can see this in brain slice experiments. If you image capillaries in brain slices and then stimulate axons which run across the cortex to release endogenous glutamate, you find you get a dilation. To see this, you have to first of all pre-constrict the vessels. Because we've made brain slices and taken them out of the brain, you've lost the locus coeruleus input, which is normally constricting them. So we pre-constrict them either with noradrenaline or, in this case, with a thromboxane analog, U46619. So they constrict, the diameter goes down, and then when you stimulate the axons to release glutamate, you get a dilation. Okay. So this dilation is blocked if you block action potentials. That's reassuring because that's how we were activating the glutamate release. It's blocked if you block postsynaptic AMPA receptors, so we think something postsynaptic is involved. But surprisingly, it's not affected if you block NMDA receptors. So contrary to many ideas in the field, this does not reflect NMDA receptors producing a postsynaptic calcium rise. So we wanted to know whether astrocytes were involved in this because Brian McVicker's group has shown if you uncage calcium in astrocytes, raise the calcium concentration, you can adjust the level of arterioles. And so we do this by patch clamping astrocytes, and we can then put into the internal solution, which goes into the cell, either a high concentration of a fast calcium buffer or a low concentration of a slow calcium buffer, so that's EGTA. And the idea is the high concentration of the fast calcium buffer will buffer the calcium concentration, stop the activation of these calcium-dependent enzymes, and thus stop the release of prostaglandin in this case, which I'm going to show you in a minute, is what's causing the dilation. So how do we do this? This shows the experiment being done. This is a brain slice with a capillary here. This is an astrocyte in the brain slice, and it's putting an end foot onto this bit of the capillary here. And after pre-constricting the pericytes, as I just mentioned, we patch clamp the astrocyte, and in this case, we put a low concentration of a slow calcium buffer inside, and when we stimulate axons to release glutamate, you get a nice dilation. Okay? You can then do another experiment where instead of putting low EGTA inside, you put a high concentration of the fast buffer Bacter inside, and then in this carefully chosen example, when you stimulate, you get no dilation at all. Okay, and on average, the dilation was reduced by about 70% when we put the strong calcium buffer inside the cell. So this tells you that astrocyte calcium rises trigger the release of something which dilates the capillaries. Now, I don't want to bore you with the whole story. I'm just going to show you a diagram which summarizes it. We believe, on the basis of a whole slew of pharmacology evidence, that when you activate neurons, postsynaptic neurons release ATP. That activates P2X receptors in the astrocyte, which raises calcium. That then activates a phospholipase, which generates arachidonic acid. That eventually generates prostaglandins, which act on EP4 receptors on the pericytes. So when neurons are active, they dilate capillaries via astrocytes and pericytes. Okay, everyone with me so far? You're all looking a bit heat-struck. So it turns out that we sent this off to a nice journal, and they said, that's lovely, but would you please do it all again for arterioles? So we did it again for arterioles, and it turns out there are different signaling pathways regulating the diameter, at least in brain slices, of capillaries at pericytes and of arterioles. So the one I just told you about involves glutamate releasing ATP, raising astrocyte calcium, releasing prostaglandins, which hyperpolarize pericytes and thus relax them. But there's another pathway. If you look at arterioles, they dilate less than capillaries. And the mechanism involves glutamate raising calcium, presumably in interneurons, to activate nitric oxide synthase. And that releases NO, which is what's responsible for dilating the arterioles. So if you block nitric oxide synthase or NMDA receptors, it totally abolishes the arteriole dilation but has no effect on the capillaries. Whereas if you buffer calcium in the astrocyte, it greatly reduces the capillary dilation with no effect on the arterioles. Okay, so what I've shown you so far is largely in brain slices. I just want to show you a few slides of what happens in vivo, because if there are any cardiovascular physiologists among you, you'll be saying, wow, what does this guy know about blood flow? It's all in brain slices. There's no blood pressure. There's no blood flow. So we want to know what happens in vivo. And to do this, we use two-photon imaging to simultaneously image the capillary diameter and the arterial diameter in vivo when we stimulate the whisker pad and look at blood vessels in the somatosensory cortex. And the idea was to see whether the capillaries dilate independently of the arterioles. If you're a skeptic, you could say that, wow, it's just all the arterioles dilating. They raise the local blood pressure here, and that pushes the capillaries open. I'm going to show you that's not the case. So this is looking down at the somatosensory cortex in a mouse in which the pericytes are red because DS red is driven by a promoter they express, and the blood is painted green with phytsidextran. And if you stimulate the whisker pad, you get a diameter increase in pretty much all of these vessels. In fact, about 60% of the penetrating arterioles, that's this one here going down into the board, 60% of those dilate when you stimulate the whisker pad. It's similar for the first order capillaries. Then there's a slow decrease in the percentage of responding vessels, dilating vessels, as you go out into the capillary branches, and that's because the pericytes further out express less alpha-smooth muscle actin. We want to look at the relative timing of the dilation of the arteriole and the capillary. And I'm going to play this movie, but this graph at the bottom shows the result. The capillaries dilate, in this carefully chosen example, several seconds before the arteriole, and so that tells you that there's an active relaxation of the capillary because it's dilated before there's any dilation of the arteriole at all. So if you look just here, this is the capillary. See, the capillary dilates very quickly, but the arteriole only dilates more slowly. I'll just play it one more time. So the capillary dilates rapidly, and then the arteriole more slowly. So pericytes are independently controlling the diameter of the capillary bed. And if you look at the numbers, you find that the penetrating arteriole dilates about 6%. The first and second order capillaries dilate a bit more, but on average, capillaries dilate about 7%. Now, what does this mean quantitatively for control of blood flow in the brain? David Kleinfeld's group have shown that they've reconstructed the topology of this part of the cortex, and they worked out the resistance if you go from the pia down to layer 4 through a penetrating arteriole, then through the network of interconnecting capillaries, and then back up a penetrating venule. And the ratio of those three resistances is 1 to 4 to 2, and the venules never change their diameter. So if you just think about the adjustable part of the diameter, 80% of it is in the capillaries. So in principle, that means it's possible that capillaries control 80% of the blood flow, and if you plug our numbers in, you get slightly more than that. So what this means is that in physiology, capillaries dilate first, and this pericyte hyperpolarization may spread back to arterioles. The capillary dilation dominates the decrease of resistance to flow in the cortex, and hence capillary dilation will also dominate not just energy supply, but also the bold response in functional imaging experiments. Okay, that's physiology. Now I want to turn to pathology. I'm going to look at the role of pericytes in ischemic pathology. So this is stroke incidence for a big city. It happens to be Melbourne, but it's probably the same in London. So it increases as you get older, which is why aging neuroscientists start to get interested in it. And 1 in 5 women in the room is going to have a stroke, and 1 in 6 men in the room are going to have a stroke. And more women have strokes than men because the men die earlier doing other stupid things, okay? Riding their motorbikes too fast, smoking more, that kind of thing. Now, most strokes involve a clot blocking an artery. So if I have my stroke here now, and you rush me to hospital, the interventional radiologist will use either a drug, TPA, to dissolve the clot, or they'll use a fancy corkscrew thing to remove the clot on the end of a cannula. And they'll image blood flowing through the artery that got blocked, and they'll say, job done, you can go away now. But it turns out the job isn't done because capillaries don't reperfuse. And there's a commonality here between what goes wrong after stroke and what goes wrong in AD. So this was shown in the 1960s in a classic experiment by Adelbert Ames III. Back then, of course, we all wrote with quill pens and ink. And so he strode over to his inkwell, and he got some ink, real ink, India ink, and he injected it into the blood of an animal, okay? And he showed that when he cut up the brain, the ink went through all the blood vessels. And then he did the same thing again after doing middle cerebral artery occlusion for an hour, and then unblocking the middle cerebral artery. Then he injected the ink and saw where it went. And he found that although the ink went back through the artery, it doesn't go through the microvasculature. So something is decreasing blood flow through the microvasculature, and that means that even after I've had my stroke and they've removed my clot, I'm still going to have an ongoing problem with energy supply, which is going to be causing me to lose neurons. And we thought we had a handle on this by imaging capillaries, in fact, in the retina. If you make the retina ischemic by applying chemical ischemia solution that blocks oxidative phosphorylation and glycolysis to stop ATP production, you find that at the location of pericytes, the diameter of capillaries goes down when you go to ischemia. And this is not surprising, because here's our pericyte with its arm around a capillary. There's lots of ATP-driven ion pumping, keeping the calcium concentration low in the pericyte. When you have your stroke, there's no ATP, so the pumping stops. And that means the calcium rises. And so the myofilaments get activated, and that squeezes the capillary. Okay, so this explains why it squeezes. What it doesn't explain is why it doesn't dilate again when the artery gets unblocked, and I'll come to that in a minute. But this also occurs after middle cerebral artery occlusion in vivo. So here what we've done is to do middle cerebral artery occlusion in a mouse where the pericytes are red, and we've got green stuff in the blood, and the blood vessel basement membrane is labeled purple with this isolectin B4. And you can see that after the MCAO, for an hour and a half in this case, and then an hour's reperfusion, blood flows back through some of the vessels, but there are other vessels, these purple things up here, which are still there, but there's no blood flowing through them. Okay? And you can assess what's happened to the diameter by drawing lots of little lines across all the vessels here using a semi-automated procedure called a graduate student. And when you do that, you can then measure all the diameters. They say it gives them a feeling of inner Buddhist calm. You find that the diameters of the capillaries have gone down more where the pericytes are. This is in the core of the lesion, than where there are no pericytes. And it also happens in the penumbra. This is all significant. So, and similar results have been obtained by these guys in Turkey. So this could, in principle, explain what happens after stroke. So this is data from other people doing middle cerebral artery occlusion where they measure cerebral blood flow. During the simulated stroke, there's no blood flow. When you open up the artery again, there's a period of rebound hyperemia. But then there's a long-lasting decrease of blood flow. So after I've had my stroke and I've had my clot removed, my blood flow in my brain is going to be depressed by about 40 or 50% for hours afterwards. So why is this so long-lasting? It's all very well to say the capillaries have been constricted by the pericytes. Why don't they open again? And I think we've resolved this in experiments in brain slices. If you image a normal capillary in normal solution for a long time, it stays at the same diameter. But if you go to ischemia, the capillaries constrict, as I've just shown you. But if you then put on propydium iodide, which is a marker of a failure of integrity of the cell membrane, it's a marker of cell death or breakdown of permeability, cell activity, you find that after an hour of profound ischemia, and this is a worse insult than a stroke, you find that the pericytes now fill up with propydium iodide. To all intents and purposes, they're dead. Okay, so we think what happens is the pericytes constrict the vessels, and if you remember your Muscle 101, or if you've seen crime scene investigation, you'll know that when ATP levels fall, you can't relax. That's why corpses are called stiffs. So they constrict the vessel, and now there's no ATP to relax the vessel again. Okay, so they constrict and die in rigor, and the vessel is permanently constricted until microglia come along and remove them at a later stage. So this is the time course of the constriction. This is the time course of death. And they're pretty much all dead after about an hour. So this was in the gray matter. The same thing happens in the white matter. So we think this rise of calcium causes the death of pericytes. Okay. And that's why you get a long-lasting constriction. So we think this is due to the death of pericytes, and that means that pericytes are a therapeutic target in stroke. We've started looking at that in collaboration with Alistair Buchan's group in Oxford. And we have two kind of start-off observations. One is that if you go to ischemia in the presence of a voltage-gated calcium channel blocker, remember that's one of the things which brings calcium into the pericytes to constrict them, then the constriction of the capillaries is delayed. And secondly, if you do oxygen-glucose deprivation, which is a more mild insult, more similar to what happens in a human stroke, and then reoxygenate the tissue, you get a lot of death in the first hour or so of OGD, and then more death in the following hour. But if you do all this in the presence of nimodipine to block calcium influx, you significantly reduce the death. And then if you mimic a clinical situation by cutting off, by doing MCAO and measuring blood flow goes down, as I showed you earlier, and it then kind of recovers a bit, but not totally. If you give nimodipine at the time of reperfusion of the artery, you find you can significantly increase the blood flow. It actually increases to more than normal, even though the peripheral blood pressure goes down. Okay? So this dilates the capillaries in the brain, and we think it increases blood flow. And this mimics a situation which could be used clinically because we're only injecting the nimodipine at the moment of reperfusion, like when you apply the TPA. And if you do behavioral assays afterwards, there's something called neuroscore, which is some measure of how well the mouse can run away from you and climb up walls, that kind of thing, you find that giving the nimodipine significantly increases the outcome for the mice after this stroke. Okay, finally, I want to turn to Alzheimer's disease. And I want to have a little digression to the properties of microglia because they may be playing a key role in what happens in what I'm about to show you. Most of what I want to show you has a very human aspect to studying what happens in AD, with some rodent aspects studied in order to understand the mechanisms. But before I get onto that, I just want to show you what microglia do. In your head, 7% of the cells are doing this every half hour. So this is a 30-minute loop. You've probably seen movies from Soyan on this kind of stuff. If you binarize the image and then add all the images together, you can see they gradually scan out the brain in a couple of hours. And when they do this, they contact other cells. Here's a microglial cell contacting an astrocyte. Here's one wrapping its arm around a neuron. And here's one, importantly, contacting a capillary. And we're interested in this because, first of all, as I showed you earlier, nearly 60% of the vascular resistance that's within the brain, which is adjustable, is in capillaries, from David Kleinfeld's work. And secondly, if you look at transcriptome studies, microglia synthesize vasoactive agents. So microglia are the only place where thromboxane A synthase exists to generate the vasoconstrictor thromboxane A2. They also synthesize endothelin, like endothelial cells, which is probably the most powerful vasoconstrictor in the body. And they also can generate prostaglandins from COX-1. So in principle, they might regulate blood flow. We don't know whether they do or not yet, although we have some preliminary data I'm not going to show today. It's known that after ischemia, they extend their processes towards blood vessels. Okay, so we wanted to know whether the same occurs in Alzheimer's disease, and it does. So if we look at the APP, NL, GF knock-in mice, which are kind of good and bad for studying AD in a way, they have a fairly mild phenotype and they don't have tau deposition, but they work rapidly and they're a knock-in so that you don't need to worry about overexpression of APP, hopefully. And if you image microglia and blood vessel and pericytes, then measure the distance simply between the cell bodies of the microglia and the pericytes, you find that in the AD mice, the microglia get closer, about 30% closer to the pericytes.
What age is that?
Yeah, that's a good question, isn't it? Normally we do all of this at P120. I could look it up and tell you later. It doesn't matter for what follows, actually. So they do get closer to pericytes, and they may be having an influence on what the pericytes do, which, as I'm going to show you, is profound in Alzheimer's disease. Do you have a question?
I was just thinking that why quite early, 4 months.
This is the NLGF mice. They start depositing plaques from about 7 weeks, something like that. Yeah. Okay. So why are we interested in blood flow in AD? You're probably aware of this study which surveyed a vast range of biomarkers and concluded that the first thing which changes in AD is a fall of blood flow. The paper's so badly written, I had to write to them to ask what was plotted, but it turns out this brown curve is the blood flow decrease due to disease with the change which would normally occur due to aging removed. So that's what changes first in AD, and that's before deposition of A beta, loss of synapses, memory loss, loss of car keys, all of this kind of stuff. That comes later. Now, of course, you can say there's a nonlinear relationship between these things anyway, which would mess up this priority for vascular, but this is not the only evidence for a vascular involvement in AD. So back in the 90s, just at the time that the A beta, the amyloid hypothesis of AD was being developed, a bunch of fairly unknown Japanese people published this paper where they looked at capillaries extracted from post-mortem AD brains. And they said, hey, look, this is really interesting. There are these focal constrictions on the capillaries. It's a kind of flaky image, but nevertheless, I think they were onto something. And furthermore, Leif Østergaard has shown, imaging both in humans and in AD mice, that there are dysfunctions of capillary control of blood flow during the onset of AD. So what he finds is that two things happen. First of all, blood flow goes down. So that's manifested as an increase in what's called the mean capillary transit time for blood. So the time, if you change the spins of some protons as they're going into the brain in the blood and measure when that change of spins comes out the other side, you find that capillary transit time is delayed. But more importantly, Leif has shown that there's an increase in the heterogeneity of that transit time, which we attribute to differential squeezing of capillaries by pericytes. And he showed that that increase of capillary transit time heterogeneity correlates with cognitive decline in AD. Okay, so these changes in control of blood flow at capillary level are already known to correlate with cognitive decline. So we wanted to look at human capillaries in AD in various ways, and we've adopted two strategies. And the core of what I'm going to talk about comes later, but this is just establishing the mechanism, first of all. So first of all, we teamed up with a neurosurgeon, Huma Sethi, to get live human tissue from patients undergoing operations to remove tumors in their brain. So when you remove a tumor from the brain, often you need to remove supposedly normal tissue from on top of the tumor. So we then collect that from the operating theater, cycle it round to the lab, make it into brain slices, and then we can do just immunohistochemistry if we want, or we can look at them live. So this is it was a live human capillary with pericytes on it. These label for classical pericyte markers like PDGFR beta, and they also label for alpha smooth muscle actin, which is what's going to be mediating the constrictions I show you in a minute. We can then look at live slices, and here's a pericyte on a live human brain slice with some red blood cells still in it. If we apply noradrenaline, the capillary diameter goes down near the cell body. I'm going to come back to this in a minute. And if you apply glutamate, it dilates. So why is it important that they regulate diameter near their soma? Why is that the case? Well, if you image transgenically labeled pericytes, you find they don't just have processes going along the vessel, they put circumferential processes around the vessel as well. And in fact, if you count the distribution of these, you find that about 80% of the circumferential processes, which are the ones that can squeeze the vessel, are within 10 microns of the soma. Okay, this is going to be important for what happens in a minute. So if you then apply a vasoconstrictor, like this thromboxane A2 analog, you get a big constriction at the soma, but a smaller constriction away from the soma. And in fact, if you plot a graph of diameter as a function of distance from the pericyte soma, you find in the unconstricted condition, the diameter is larger near the soma. We think that's because the pericyte induces growth of the endothelial tube. But after you put the constrictor on, you get a big constriction at the soma. And we're going to see this in a minute manifested in AD. So the percentage constriction is largest near the soma. So just keep this diagram in mind. So what's the relevance to the control of blood flow in AD? Well, if we use a standard protocol to make A beta oligomers, they're mostly about 4 times the molecular weight of the oligomers are about 4 times the molecular weight of the monomers. Then when we apply A beta 1 to 42 or 1 to 40, we get a constriction of the capillaries. Now, this is on a very rapid time scale. You're probably thinking this has got no relevance to AD because AD happens over decades, and this happens over an hour. But I'll try to convince you otherwise. To look at the mechanism, because we can't get much human tissue, we turn to rodents. So here's a rather jerky movie of a pericyte squeezing a capillary in a rat in response to A beta applied now. You can see the diameter gets smaller. And you can pick stills off this kind of movie and plot graphs. And what you find is that both 1 to 42 and 1 to 40 constrict capillaries, whereas scrambled A beta, which doesn't form oligomers, incidentally, doesn't constrict them. And if you look at the dose-response curve of this effect, you find that EC50 is about 4.7 nanomolar, measured using the monomeric molecular weight. And Colin Masters' group has shown that if you extract soluble A beta from AD brains, the soluble concentration is about 6 nanomolar, which means that this is in the right dose range to have an effect in real AD. So what's the mechanism? Well, there's a whole slew of receptors which have been proposed as proteins which have been proposed as receptors for A beta. I'm not going to get into which one is doing it, but the general opinion is that when A beta oligomers bind, they activate NADPH oxidases to pump electrons out across the cell membrane. And so it's interesting that this constriction we see generated by A beta is blocked if you block all NADPH oxidases with diphenyliodonium. The electrons here usually make superoxide. So it's interesting that we find that superoxide dismutase also completely blocks this constriction. And it's previously been shown in transgenic mice that if you overexpress SOD1, that also decreases the death, if you like, of AD mice. And then finally, if you block endothelin A receptors, and this was suggested by previous work in the literature, partly by Colin Masters' group, showing that peripheral blood vessels can be constricted by A beta in a manner which depends on endothelin release. And also Seth Love's group has shown that the level of endothelin goes up in the postmortem AD brain. That also completely blocks this constriction. So we think somehow the reactive oxygen species release endothelin, and that acts on pericyte endothelin A receptors to constrict them. At the moment, we're not sure which NADPH oxidase it is, but I'll show you some suggestive data that it's NADPH oxidase 4 in pericytes. All of these cells express endothelin, so we don't know where the endothelin is coming from yet. So all of this is happening. Why have I put the reactive oxygen species before the endothelin? Well, if you apply endothelin on its own, it constricts, as I showed you earlier, and that's not affected by superoxide dismutase. Whereas if you apply reactive hydrogen peroxide, generates reactive oxygen species, that constricts, but that constriction is partly blocked by blocking endothelin A receptors. So we believe the reactive oxygen species come before the endothelin. And with more pharmacology, we find that blocking NOX4 blocks the constriction totally, whereas blocking the microglial or endothelial NOX2 only blocks it by 45%. So we think it's NOX4 that does it. And we can look at this directly by imaging ROS production using dihydroerithidium. So when you put A beta on, some cells go red, and this is blocked by the SOD1, as you can see here. And if you image the cells in more detail, you find the cells which are going reddest are pericytes, which are labeled with Ng2, whereas the immune cells don't go so red. And quantifying this, immune cells double their ROS production in response to A beta. This is in the short term. It may be different in AD in the long term, where the timescale is completely different. But although immune cells double their ROS production, pericytes increase it by about a factor of 6 or 7. So we think pericytes dominate the process. Now, all of this is very short-term application of stuff. So how can you be sure this is relevant to real AD? Well, it turns out that if you're dementing for no known cause and you happen to be walking past Queen Square, they will happily, if they think there's a possible clinical benefit, remove part of your brain while you're still alive from the right prefrontal cortex. It's a non-eloquent area. You won't notice it at all, sir. And they then look for A beta and tau, or whether there's some other cause of the dementia they can deal with. They then archive the tissue, and we can then access that tissue. They only do this once or twice a year. We can then access this historical tissue and look at the pericytes. And so we got access to this tissue, and with the help of Sebastian Brandt and Zane Jalmuktani, we labeled them for PDGFR beta. Half the patients have no A beta deposition, so they're not getting AD, presumably, and the other half do have A beta deposition and also tau, and so presumably they are getting AD. And we can look at the pericytes, as you can see here, and the capillary diameter in these two classes of patients. And you then get that graduate student again to measure thousands and thousands of capillary diameters in these images. And on average, you find the diameter's gone down by 8% in the AD tissue. And you say, well, that's okay. It doesn't sound like much. Although by Poiseuille's law, this will reduce blood flow by 30% if it was the same everywhere. In AD patients, the measured decrease of blood flow in affected areas is about 40%. So it's the right ballpark. We wanted to know whether this is just some non-specific change or whether it's due to the pericytes. And I remind you, I said earlier, Seth Love's group showed that endothelin levels go up in the AD brain, and those Japanese guys showed these focal constrictions as well. And so to pin this on pericytes, we simply measured the diameter as a function of distance from the pericyte soma, like I showed you earlier in the brain slices. And when we do that, we get a graph that's very similar to what we saw in the brain slices. In the patients with no A beta deposition, the diameter is larger at the soma, and I attributed this earlier to pericytes inducing growth of the endothelial tube. But in patients with A beta deposition, there are just 4 points here, but there's really 5,000 points behind this line, which is why it's so massively significant. There's a massive constriction at the soma, okay? So I remind you of this graph I showed you earlier, just applying a constrictor in a brain slice. That's very similar to this diagram we've got in the human AD patients, okay? And this predicts, if you take these two profiles and do the maths, for Poiseuille's law, that predicts a 50% decrease in blood flow. We can then look at patients with different degrees of A beta deposition as assessed by the neuropathologist, and you find that the slope of this line goes from negative down here to positive smoothly as you go from no A beta through moderate to severe A beta deposition. And more scientifically, you can quantify the amount of A beta optically. I did this just by processing these images to try to measure the amount of brown in the image. And you find then that this extrapolates, each dot here is one patient. If you draw this graph for each patient and work out the diameter at the soma, as the amount of A beta deposited goes up, you find that there's a very rapid decrease in the diameter at the soma of the pericytes. So the strength of this constriction increases with the disease. So we sent this to a nice journal, and they said, that's all very well, but have you thought about doing it in a mouse? So we hadn't. So we did, and it's the same. So this is, again, the APP-NLGF mouse. The mean diameter goes down, the decrease is larger at the soma, and again you get this nice profile of larger at the soma in the wild type, smaller at the soma in the AD mouse. And you can, in the mouse, you can look at the cerebellum, which is a region which at this age, which is about 4 months, it's funny none of these, here we go, here's the age. At this age, there's no A beta deposition in the cerebellum, and also you don't get the constriction. Even the ones, the AD mice, don't have a constriction at the soma in the cerebellum. Some people have claimed that exogenous A beta constricts arteries. For example, that it constricts the aorta. So we looked at arterioles and venules, but they're not changed. So we think that all of the decrease of blood flow, at least in the mice, we can't be sure about the humans, is generated by pericytes constricting. Now, a paper came out recently by Chris Schaefer and Nozumi Nishimura's group claiming that in AD mice, they get a block of capillaries, which is due to neutrophils blocking at certain sites in the capillary. They didn't correlate this with pericytes. We assume, so they found that the percentage of blocked capillaries went up from 0.4% to 1.8%, which is they predicted to reduce blood flow by 5%. Our constriction, ignoring that block, is predicted to give you a 50% reduction. So we think that neutrophils may come along in the blood and get trapped where there's a constriction produced by the pericyte. This remains to be shown definitively, though. Right. Can we prevent or reverse the constriction? I think I'm running out of time, so I better go through this quickly. Therapeutic approaches. I've shown you that this is the pathway by which A beta works on pericytes. NOX4, possibly in pericytes, generates ROS, releases endothelin. That activates endothelin receptors, which then dumps calcium from stores. There's this amplification, and you get a constriction. So one thing we might do is block NOX4 or block endothelin A receptors, but the problem with endothelin receptors is they're incredibly high affinity for endothelin, and in fact it's believed that it's very hard to reverse this constriction once it's started. The action of endothelin is actually terminated by the whole receptor with the endothelin bound being taken into the cell for degradation or recycling. But anyway, we'll try it. But then, as another approach, we wanted to try a C-type natriuretic peptide, which you've probably never heard of, but if you've ever done cardiovascular physiology, one of the things which maintains your blood pressure and volume is atrial natriuretic peptide. It controls sodium loss from the body, and it has a relative which begins with C. This activates a guanylate cyclase receptor, which acts through this protein IRA-G, which stops calcium release from internal stores. And at the same time, a bit like Viagra, I think, it raises the cyclic AMP concentration, and that activates myosin light chain phosphatase, which stops contraction of the myosin and actin. So we wanted to try both of these. This is what I've just shown you. We activate a constriction with A beta. Here's the constriction normally. If we block this activating pathway, it stops any further constriction, but any recovery is clearly going to take a long time. But when we apply the CNP, you get a rapid reversal of the constriction. So we now think that we need to stick with the CNP and try to find ways to target to pericytes in the AD mice initially. So this is the core of what I've told you today. A beta constricts pericytes. That decreases blood flow and decreases energy supply. This reconciles genetic evidence for the importance of A beta production in AD with the fact that the earliest event in AD is vascular disruption. I've shown you some therapeutic possibilities. Now, it turns out other people have shown that there's a positive feedback loop here. When energy supply goes down, you upregulate BACE, the enzyme which generates A beta, and so that's going to make more A beta, and that's going to cut off energy supply more, and you're going to go round and round this circle. And what that means is that the A beta and possibly downstream tau, which are produced, are going to be amplified by this amplifier, producing synapse and neuron loss. But it also raises the possibility that if you suffer a blow to the head, or if you suffer sleep apnea, anything which decreases your energy supply to the brain may kick off this very slowly developing cycle here. Finally, and provocatively, I want to suggest that AD is a vascular disease predominantly. I want to raise the question, what percentage of the loss of synapse and neurons is produced by A beta and tau? and what percentage is produced by the loss of energy supply, which A beta ultimately produces. These are the people who did the work. The AD story was led by Ross Naughtley and Niels Korter. The physiology was led by Anusha Mishra and Catherine Hall. And the original physiology was done by these guys at the bottom here. Thank you for listening.
Thank you, David, for the fascinating talk. Now we have a few time for questions. So,
Thanks for a really great talk. I had a couple of questions about the relationship between the CA and sort of the A beta deposition and capillary constriction. So the first was when you looked at the relationship between plaque A beta and constriction, did you also look at CAA, so amyloid deposition in the vessels, and whether either the severity of this or
Yeah, there wasn't, in the patients that we were looking at, there wasn't that much. I mean, some of them had some CAA, but all we correlated it with was A beta and tau. A beta and tau correlated perfectly, pretty much. So there were patients who had no A beta and no tau, and then all the other patients had kind of parallel variation of A beta and tau. I think it's quite a small number of patients, it's 6 or 7 in each group, but in those patients, it may just be that they went to hospital to have their biopsy early enough that they hadn't got much CAA. The mechanism that I've shown you with the short-term application of A beta and the endothelin may not need CAA to occur. So I kind of think of CAA as a mechanism which damages pericytes late on, and if they haven't already constricted and messed things up, they may then die and you get blood-brain barrier breakdown. But I don't think it's causal in this.
So do you think they're recoverable, just thinking about the fact that amyloid immunotherapies cause this transient increase in CAA?
I think that, sorry, say that bit again?
Do you think the pericytes are sort of, if they get damaged by CAA, do you think that's recoverable? Just thinking of all the therapies we're developing that are amyloid immunotherapies that cause a transient increase in CAA.
Right. I don't have a view on that, but I think that treating AD in the long term is going to be a bit like treating high blood pressure. So, half the people in Britain over 60 are on blood pressure modifying drugs, and I think it's going to have to be like that for AD. And it may be that, one thing I'd like to do is to do some kind of epidemiological, if that's the right word, study. Half of those people on blood pressure modifying drugs are on voltage-gated calcium channel blockers. And I would predict they're going to get AD later because their pericytes can't constrict so much. There's a paper already in the literature. And the problem with giving calcium channel blockers, both for stroke and for AD, has been that doctors will always say, oh, well, the peripheral blood pressure is going to go down and you're going to decrease perfusion. But in fact, there's evidence in the literature this is not the case. There's a study with, I think it's called nilcardipine or nilvardipine, showing that after ischemia, in AD, although peripheral blood pressure does go down, you increase brain blood flow when you give a voltage-gated calcium channel blocker. So I think it's quite a good idea to do some kind of study, getting patient data, and looking at whether the ones who are already on voltage-gated calcium channel blockers from an early age, because it's no good giving it late, either don't get AD or develop it later.
Thanks for the talk, Dave. So, in terms of the pericytes, you were looking in barrel cortex a lot, but do you think that this could contribute to the region specificity involved in Alzheimer's disease?
Our functional experiments in the mice were in barrel cortex because that's what people study. So it's easy to get, you can stimulate the whisker pad to get an evoked response. The data on the humans were from prefrontal cortex. I don't have anything to say about the order with which things spread through the brain in AD. I think that's a different question. Ultimately, A beta is producing the decrease of blood flow, I think. But again, that remains to be shown in an in vivo human situation on that very long time scale. You'd think there'd be some kind of compensation, but all the parts of the mechanism are there. Certainly the pericytes are constricted, the endothelin has gone up, the A-beta's there. Yeah.
I'm a bit confused about the nifedipine experiment in the ischemic cases. So the pericytes die in a kind of rigor mortis. And so you showed that you could rescue the phenotype by nifedipine. So, I mean, and you showed that this dying of these cells is very early, in fact, within 1 hour or 1.5 hours.
Yeah, hang on a sec. That's with profound ischemia, when you do chemical ischemia in a dish. When you do MCAO in vivo, they die much more slowly. They only start to die after 24 hours or so. So the behavioral experiments I showed with the neuroscore, that was done giving nimodipine after the ischemia, not during the ischemia. So you're rescuing it by giving it when you would reperfuse. And you got a better outcome, but they weren't perfect.
And so why do they die, basically?
They die as a result of calcium influx. In our original 2014 paper, we do loads of pharmacology on why they die. And we thought it was going to be reactive oxygen species, but it doesn't have any effect. Nitric oxide has no effect. The only thing we found that stops them dying is taking the calcium away. So something downstream of calcium.
So I find the timing is a bit interesting. Like you can have, so at the early stage, even a low soluble concentration of amyloid beta can cause like vasoconstriction and stuff. But in terms of actual cell death, it only normally happens like late stage with like Alzheimer's disease. So
yeah, I'm not saying these cells were dead. I don't think the cells are dead that we saw in the biopsies. I think they're just actively constricted.
So you mean it needs a long time for that blood flow to contribute to cell death in the late stage?
Are you talking about neuronal death?
Neuronal cell death.
Right, I assume it takes 10 or 20 years for the A-beta concentration to rise high enough to gradually decrease the blood flow and lead to problems for the neurons. That's what I'd assume. Bart wants to say something.
Yeah, I have also another question about the mechanism. So
Sorry, so I've got a complaint from a YouTube listener that he cannot hear questions. So I'm so sorry.
Oh, I'll
Do
say
you
them
mind
again.
quickly rephrasing the question that they asked?
Okay.
Okay, good. So about amyloid and the vasoconstriction. So, I mean, in the patients, you talk about amyloid plaques. And in the experiments, the physiological experiments, use soluble A-beta. And so I think there's a fundamental difference between the two. Soluble A-beta can interact with receptors, etc.
Okay, I need to repeat this question, so keep it short. So the question is, why are we thinking about amyloid plaques in the patients and versus soluble A-beta in the short-term experiments? And
But actually, the question I want to ask is, don't you think that with amyloid plaques you get another mechanism than the one you showed with the soluble A-beta? And I can explain why I think so.
I view the plaques as a reservoir of oligomers. So in the patients, it's fixed tissue. All we can do is label the plaques. And we assume that that's a marker for where the A-beta oligomer level would have been raised locally. We're now doing experiments in the mice where if you look at the mice, some of them have capillaries blocked as A-beta starts to be deposited. And we're measuring the distance. So in brief, there are more capillaries blocked near the plaques. Okay. But we attribute that to the A-beta oligomer level being higher near the plaques. Yeah.
Sorry, another one. Just related to that, do you think that A-beta peptides of different lengths would influence the mechanism here? And have you looked at longer A-betas like A-beta 43? Because we see in familial Alzheimer's disease that mutations that produce a lot of A-beta 43 seem to have particularly severe CAA.
Okay, so the question is, why have we only looked at A-beta 1 to 40 and 1 to 42? Would longer A-betas have a similar effect. I don't know whether it would. I'm well aware of the fact there are all these different species which may be having an effect. We've only done 1 to 40, 1 to 42, and the scrambled A beta. The scrambled A beta we couldn't oligomerize, and it had no effect on the diameter. So I believe that it's formation of oligomers which is necessary to bind to whatever receptor it is to trigger all of this happening. But it would in principle be easy to test that if you want us to, if you can give us some. If you can oligomerize it and bring it over, we can dump it on and see what happens.
A question from one of the audiences, Sadia Hassan. She's asking, do you foresee this mechanism of neurodegeneration due to pericytes? In your case, it's very A-beta driven. Pericyte-induced vasoconstriction in AD translate to other forms of neurodegeneration, such as PD, ALS, and FD, if so how?
Yeah, I think it will, but we're going to look at that in a synuclein mutant mouse. So basically anything where reactive oxygen species is produced, if you believe this mechanism, you would predict that that's going to release endothelin and cause constriction of pericytes. So we now, it's in my grant application, we would like to get synuclein mutant mice which have increased ROS production and see whether in the parts of the brain that are affected, you see selective pericyte constriction. So I think it will extend to HD and PD.
Right. And I saw that my question is now I see that you you're looking. Are you planning to look at TREM2 knockout microglia to see whether that plays a role in this parasite?
Yeah. Well, we have the TREM2 knockout. Yes. Yes, we are.
Yeah.
Yes.
And another question relating to vulnerability, so selective vulnerability. So that's a big field, big idea in the field that there is this remarkable region specificity in different neurodegenerative diseases, right? So is there anything known about parasite heterogeneity and is between, for example, between gray matter and white matter? And could you speculate on if such heterogeneity exists, how they could contribute to the selective vulnerability that we see?
Yeah, there probably are differences. There's a whole slew of transcriptome data available, all of which is mutually contradictory, so it's hard to say anything on the basis of that at the moment. But I think another kind of heterogeneity is between humans. Some humans, for people, if you take a pool of people, a lot of them will have high A beta or have plaques, but they won't have any problems. And so clearly for those people, maybe there's been an upregulation of some compensatory mechanism which stops all this happening. And I think we need to try to figure out what it is about those people. And I'm not sure whether it's possible to look at that in the mouse line or whether the mouse is too kind of inbred and homogeneous. But that's certainly, if we could get a handle on that, that might produce other therapeutic approaches.
Yeah, that'd be really great. Is there any other questions? No? Done? Thank you so much, David.
Okay, thank you.
I was really just thinking out loud because I remember it'd be really interesting to see if microglia